Hello, my name is Laura. I'll be talking about gene expression regulation with eukaryotes today. Remember that structural genes encode proteins that we're regulating. The regulatory gene encodes RNA or proteins that control structural genes. Regulatory elements are unexpressed sequences of DNA. They don't produce the RNA and protein, and these unexpressed sequences can affect the expression of structural genes too. Regulatory genes, because they produce a product, their product can move around in the cell and they can act on either pieces of the DNA. This makes them trans. Regulatory elements instead are cis because they can only act on the piece of DNA they are attached. For galactose metabolism regulation in eukaryotes, the structural genes here that are being regulated are Gal7, 10, and 1, and these, when produced, reduce galactose. They break it down. And these are controlled by Gal4 and Gal3 regulatory proteins. Gal4 is inside the nucleus bound to the DNA, and the activation domain turns on structural gene expression to break down galactose. Gal3 is in the cytoplasm and binds to galactose. So Gal80 is the thing that controls both 4 and 3's activity. So when there's no galactose, Gal4 will be bound with Gal80, and the activation domain will be halted. It will be inactive. So the expression of 7, 10, and 1 will be stopped, and galactose will not have to be broken down. When galactose is high, Gal3 will bind to galactose, and Gal80 will bind to Gal3. This means that Gal4 is free, and the activation domain is active, and Gal7, 10, and 1 are expressed. If Gal7, 10, and 1 are expressed, this means that galactose metabolism will occur, and galactose will eventually decrease in the cell. So what is the effect of creating a mutation of the galactose binding site of Gal3? So if Gal3 can't recognize galactose, Gal80 can also not recognize galactose. So Gal80 won't bind to Gal3, it will always bind to Gal4. So D is correct. So if Gal4 is always bound, the activation domain is not working, and the structural genes to break down galactose aren't being produced. So galactose will eventually rise in the cell because the machinery to break it down isn't being created. So the correct answer would be A and also D. So E, 2 of the above are correct. So what is the effect of an early frameshift mutation early on in Gal80's regulatory gene? This means that the reading frame is shifted when Gal80 is being translated into a protein, and so Gal80 isn't really being created as Gal80. It's eventually going to be non-functional A because you're not producing the same amino acids that you were if you had the correct reading frame. So if Gal80 is non-functional, A is correct. This also means that Gal80 cannot bind to Gal4, and Gal4 can always create the machinery to break down galactose. So D is true, and the correct answer is F, A, and D. So also, what is the effect of receiving a drug that acts like galactose and permanently binds to Gal3? So this is similar to the first question, but a little bit different. This drug is binding to Gal3, mimicking galactose, but it's not letting go. So Gal80 will find Gal3 and this galactose-like molecule, and it won't let go. So this means that Gal3 will always be open, and if Gal3 is always open, it's always active, and triggering the expression of the structural genes that are responsible for breaking down galactose. So the correct answer here is A. So moving on from galactose, androgen. Remember that androgen receptors bind to androgen coming from outside the cell, and once bound, they form a dimer in the cytoplasm. And so this dimer can bind to the DNA at the response element and activate the expression of many other genes. So what is the effect of mutating the DNA binding site of an androgen receptor? This doesn't necessarily mean A, that the androgen will not be able to enter the cell because it was able to enter the cell before it bound to the receptor in the first place. This doesn't necessarily mean B, a dimer will not form because we're mutating the DNA binding site, not the dimer binding site. It can't be C either because eukaryotes don't have operons, those are only in prokaryotes. But the answer is D. Multiple genes are not expressed from many different promoters. And this is because, number one, there's not operons, and that the androgen dimer, if bound, does induce an expression. So if it doesn't bind, the expression is halted. All right. I hope this video helps for your exam 3 and understanding a little bit more about eukaryote regulation.